You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Dr. Jonathan Clayton. He's an assistant professor at University of Nebraska, and we're going to be talking about host-microbiome interactions in humans and non-human primates, which is super fascinating. So, Jonathan, thanks for coming.
Yeah, thanks for having me.
Yeah, I saw that your moniker is the Monkey Doc. Why is that, by the way?
Yeah, so I guess that came to be over a long history, really. So my interest has always been in non-human primates for a very long time, and ultimately I decided to go to veterinary school initially, and did finish veterinary school. But that kind of came to be during veterinary school because my passion, even once I entered veterinary medicine and primates, is obviously, well, in the context of veterinary medicine, it's very specific. So it came to be kind of that way. It would just sort of kind of came together during veterinary school and just kind of kept it ever since, and got a website and things like that, and just kind of used it as a small trademark, if you will.
You said you're studying host microbiome interactions?
Yes, sir.
So I've heard the term holobiont put around, or superorganism or metaorganism. Do you see it as a host and microbiome interaction, or do you see it as a necessary whole, meaning that our microbial constituent makes a creature what it is, and without it, there is no creature? Or do you see it as really two separate things that are interacting?
Sure. That's a very, very interesting question. So do you mean in terms of you can't have one without the other, or?
Yes, exactly right.
Right.
Or you can have one but barely alive without the other.
Sure. Yeah. So I would argue that you can't really have one without the other. I mean, and I can explain why I think that, but one exception would be, so there's these things called notabiotic mice or germ-free mice. So germ-free mice are essentially the purpose of them in the context of research are they're used for various different experiments, particularly when you're looking at the microbiota. And the reason for that is because, we have other mouse models that we've controlled genetically, for certain types of cancer or whatever it is that you're studying. But in terms of microbes, we have so many microbes in our guts and on our bodies, but especially in our guts, that it's a very complex system to study, particularly if you're looking for some type of phenotype, let's say, like what bacteria causes what. And so germ-free mice, the point is that germ-free organisms or things that are basically reared in, basically in sterility, they don't do very well. They do live for a period of time, but not really. Their immune systems don't develop, and so on and so forth. And then when you try to take that to larger organisms, like for example, there are germ-free pigs, but they also don't do very well. So there's a lot of evidence to suggest that we need our microbes. So and the thing is that we know that we've basically co-evolved with microbes, but it's definitely a paradigm shift in thinking about things. But I certainly believe that without our microbes, we wouldn't have, our immune systems wouldn't be primed. We wouldn't be able to digest many of the foods that we consume. And then a whole host of, we wouldn't be able to protect from pathogens. So they basically do it all. And then another big thing, I mean, I don't want to throw out a statistic or a number, but microbes are also extremely important for drug metabolism. So many of the drugs that we take actually require microbes to actually turn those drugs into their active component.
If you observed, or people talk about the microbiome, they always talk about the gut microbiome. But I've spoken to a number of scientists. I mean, just one today, they said the pancreas has its own microbiome. Tumors that may form in the pancreas have their own microbiome. It seems like an entire monkey or an entire person, every part of them is covered in different microbial colonies. Every organ, every part of their skin, the whole creature throughout is as microbial constituents. I mean, is that what you're thinking, or have you not seen that?
Yeah, no, I mean, that's a very good, another very good question, very good point. Yes, I do believe that, that basically we have these microbial communities that are site specific, if you will. So there was the famous Human Microbiome Project kind of really elegantly showed that they were looking at 18 different body sites, but they showed that amongst those various different body sites, even if, let's say, you go from the armpit to the elbow, it's a very different microbial community structure in those two micro environments. So even on the skin, let's say, for example. So and in terms of I'm not a cancer biologist, and I don't really work with tumors, but I've worked with people that do, and I've heard a number of talks that talk about the microbiome of the tumor environment, as you said. So that is a promising area for cancer research, that understanding that when a tumor forms, it actually does have its own unique microbiome. So again, the association potentially between the microbes and the either the development of cancer or whether it's a scaffold. But yes, in short, I do agree that microbiomes are site specific.
So in studying the host microbiome interactions, what are you studying? What have you been observing?
Right. So primarily our research has focused on host microbiome interactions in humans and non-human primates. Some of the work that we have done previously, or one of the sort of landmark things that we studied and found out was that primate dysbiosis. So dysbiosis meaning deviation from normal. So this is an abnormal microbial community structure, and it's basically just a term for abnormal, and it's usually associated with being with an unhealthy microbiome. So we discovered that one of those things is that's driven by a loss of, in primates at least, bacterial diversity, and that is not groundbreaking or anything, but we showed that in both wild and in captive primates. But specifically, we showed that as you move from we used basically non-human primates as a model system for studying the effects of immigration and lifestyle disruption on the human gut microbiome. So basically what we did is we took two model different non-human primate species, both in the captive and in wild. So these were red-shanked dukes and mantled howlers. And it's not as important that what species they are, but basically what we showed was that these different primate species have very distinctive signature microbiota in the wild. But once they're in a captive setting, they actually start to lose their native microbes and become colonized with bacterial species that are dominant in the human microbiome. So in our particular study, these wild monkeys, these wild primates didn't have any of these bacterial taxa that are dominant or genera that are dominant in the human gut microbiome. But all of a sudden, when you under captive settings, they basically have a drastically different microbiome, and that microbiome more closely resembles humans than their wild counterparts. And that's why we titled the paper "Captivity Humanizes the Primate Microbiome." That's one of the things that we've been, yeah.
What have you found? Does that change them phenotypically? Like, I don't know, has anyone even tried to correlate that?
Correlate it with.
So when a primate is now captive, you said their microbiome changes over. Like, over what period of time does that happen? And then what happens to the captive? I mean, does it change its behavior dramatically? Does it change its its outer appearance, gene expression?
Yeah, no, I think that's an excellent question, and that's really the next set of, or at least for some of the next set of experiments that need to be done. I mean, some of the challenges are for this type of work is that a lot of these, if you take, for example, now we're not bringing in wild primates really into zoos and stuff like that, not wild primates. So you work with, let's say, a sanctuary or something like that. So there's just a lot of infrastructure and things to set up to basically catch, for example, that moment when that primate, let's say, goes to a sanctuary or goes to a captive setting, because really what we have seen is that, or what we do know, or at least we think, is that the change happens relatively rapidly. So whether that's diet, whether it's environment, whatever it is, but for example, these howler monkeys in Costa Rica, this sanctuary is very approximately located to actually where the wild individuals are. But what we see is that we actually had one individual, interestingly enough, that we sampled, and it didn't cluster with the other captive individuals. And when we went back and looked at the data, what we determined was that individual had only been in captivity for 2 days. So it's most likely that in a week or 2 weeks, and we don't know the exact timeline, that microbiome would have shifted to be with those other captive individuals. So the point being that even in a sanctuary that borders the wild individuals, their microbiomes change rapidly once they enter a captive environment. But yes, we are trying to do studies to try to basically zoom in on that and try to understand, one, what the timeline is, like how quickly does it change, and then second of all, what does that mean in terms of, let's say, health or whatever. I mean, we make the assumption that wild primates are, I guess, if you will, healthier than captive ones. I mean, that's not necessarily true, but in certain species that are very difficult to keep under captive conditions, which was the case in this particular study, we can kind of allude to that. We don't say that, it's not a smoking gun, and we can't say that for 100%, and it's correlative, but we assume that's the case because we know under captive conditions, these, for example, these primate species, they don't thrive per se.
So maybe a big component of them not thriving is that their microbiome fraction changes to the point where it doesn't work well with their body. It's so different to them, that causes problems. Right,
exactly. And then it also, that's exactly right. And it could be too, it may be different if you were, example, transitioning, a primate from a wild setting to a captive setting versus an individual that was born in a captive setting. And that's all that's all it ever known. There may be something to that too. And again, just because, there's a lot of logistical and study design things but we are working to try to set those studies up, and we do have some stuff running right now where we're trying to investigate that actually.
Have you tried to release any primates into the wild, tag them and capture them a month later?
Yeah,
see if their microbiome went back?
Right. Another great question. No, we have not. But again, that's something that we would like to do. Most likely we would do it non-invasively, in the sense that we would track them after they're released and see what their microbiome looks like. But then re-bringing them back in, that's an excellent question. We have not done that. That would probably be more opportunistic, because in those particular cases, we'd have to have good justification, good reasoning for doing that, and then of course a lot of permits and various things like that. But it's an excellent question, and it's I think the important thing what you're highlighting is that it's also a basic science question. It's a question that we don't know, and part of that is just a logistical issue, which again we're trying to work through.
So when you talk about host microbiome interaction, what kind of interactions have you looked at or characterized?
Yeah. So again, we've been focused on the relationship with diet, and in particular fiber content on the microbiome. That's one area of focus. Another area of focus that the lab is currently, or my lab is currently focused on, is looking at the gut brain axis, or the gut microbiota brain axis. So there's a lot of evidence to suggest that there's a very strong relationship between we know there's a relationship between the gut and the brain, because we know that how innervated the gut is in terms of it's very, there's a lot of nerves that, and particularly the vagus nerve, which is a cranial nerve. So we know that bacteria can produce neuropeptides, things like serotonin, for example. So there's a lot of interest in this particular area of research, and so we're using a marmoset model that's a small New World primate to investigate this and further. So we're trying to understand the relationship between stress and the microbiome. So how when an individual is stressed, how does that change their microbiome? And then when an individual has dysbiosis, does that result in an individual being stressed?
Well, how do you define diversity? Does that mean that certain species are not present, or that certain species come to dominate disproportionately? And same thing with dysbiosis. What is dysbiosis? And
yeah, exactly. Yeah,
how do you know if you don't compare to normal?
So that's another lot in that, but another really great question. So how do you define dysbiosis? I mean, again, you're exactly right. In order to do that, you have to know what normal is, for sure. So I 100% agree with you there. I think that, and that's why studies that are not basic at all, but studies that are characterization based, in the sense of, we take, so-and-so healthy individuals, we follow them for this period of time. We really drill in and look at exactly what are they eating, how are they living, what are they doing. They're, again, very hard studies to perform because they're very, you have to have a lot of compliance, obviously. But those are some of the studies that we're starting to do in the monkeys too. But these are exactly what you're saying. These are what gives you that baseline data then to be able to determine, what's abnormal. And the other piece of that that's important, and actually this was one of the reasons why the Human Microbiome Project was so important, is because one of the things that the Human Microbiome Project showed is that there's a lot of individual to individual variation. And so, when in that particular study, basically the initial cohort of individuals that they recruited were healthy individuals. So that was one of the criteria. So these healthy individuals, when you just look at this set of healthy individuals, between individual inter-individual variation or whatever is super high. So to put in a nutshell, basically, one individual's microbiome that's healthy, another individual's microbiome is healthy, but they're very different. So then it's hard to just basically take that, compile that, and say this is what a healthy microbiome is. It more and more evidence is suggesting that it's very host specific and then individually specific.
Well, I'm sure it's host specific, but there has to be hallmarks beyond just a blanket word, diversity.
Oh, I see exactly.
Tell you what's going on.
I see what you mean. Yeah, sorry, I missed that part of the question. No, absolutely. So I think a few things there. One is, yes, you're absolutely correct. Diversity has been used as a marker for health. I think that's because it's one of the few things that was present, and also came from studies like where we're looking at healthy versus unhealthy individuals, and the healthy individuals had a lot more higher diversity than the unhealthy individuals. But again, that's absolutely obviously not perfect, and that doesn't because one of the areas that is getting pushed to now is looking at the metabolism. So for example, let's say we have 10 different species of a bacterium, but those bacteria all perform the same function. The fact that you have those 10 different species, it may matter quantitatively how much all those collectively are in terms of numbers. So numbers definitely matter. But when we're talking about diversity, we're not talking so much about numbers. We're talking about numbers of different taxa. So I agree with you that has been used as a marker, but that may not be perfect, because really it's more about what the bugs are actually doing in the gut. And if you have a number of bugs that are performing the same function, it really doesn't matter how many different bugs. It's more about what the bugs are doing. Does that make sense?
Well, it does, but I mean, for instance, I wanted to do this experiment on myself. Is
sure.
Sample my oral microbiome a couple of 100 times over the next year, before I eat, after I eat, when I wake up, when I go to sleep, when I'm healthy, when I get sick. And at least that takes away the variation, between me and other individuals. And I can profile my microbiome maybe and see what does it look like when it's healthy, and then when I get sick,
right,
or under different conditions, what's it doing?
Right.
And then maybe that would give me a clue, or, do this for one of the primates. Maybe that would give you a clue as to what the bacteria are doing, because you'd see their absence or presence, or, waxing or waning under different, physiological conditions.
Oh, that's absolutely correct. Like, you're dead on. And I think you alluded to maybe what I didn't articulate very well, but that's the fact that we need these longitudinal types of studies. So, for example, sampling at, 1 or 2 time points, particularly when you have a lot of, or just even one time point, it's really not all that useful. I mean, I guess it definitely depends on the situation, depends on the study design, it depends on the, and a lot of this is obviously opportunistic in most microbiome studies, but But exactly right. You have to have a longitudinal component because you have to be able to then, determine one, you have to be able to basically get rid of the noise. So you have to understand that the microbiome, while it is relatively static, it's also dynamic. And so it's going to change over time, no matter what. And so in order to then determine whether or not, let's say it's your change is because you took an antibiotic or it's just normal change, you have to have those multiple time points as you're referring to in order to elucidate that. So yeah, I agree 100% that is the way that you get to the answer to that question.
Are you able to do these? I mean, even if they weren't very longitudinal, why not take, 3 marmosets and sample them, over the next week under 10 different conditions?
Yeah, no, that's
maybe they'll give you a quick view of how much change is there.
That's absolutely what we're working on that right now, and that's a great point because even for example, in the particular colony that we're working with, we're planning to do that with every single individual. So very micro scale time points and follow these individuals to just have that baseline knowledge of what is how does it change, what's normal, what's abnormal, and obviously that's going to help us when we ask all these other additional questions.
What's interesting is I wonder how the host and its microbial constituents shape your immune system, because the immune system, for instance, already it's a collective. It's like a police force, at least for our somatic cells. But I have the suspicion that it's also informed by our microbial constituent, and the immune system is like a crossover between the two. The microbes are inside of us so they can live and thrive where they're host,
right?
Essentially. So I would think the immune system is very important to them in maintaining their environment, so they would play a part in it and communicate with it,
right?
Modulate it.
Yeah, 100% agree. I mean, obviously, when you're talking about the immune system and you talk about the microbiome, both of these types of data are, complex, pieces of data because there's a lot of things at play. But absolutely agree that they're. And this is, accepted that the microbiome and the immune system are intimately linked. And in particular, when you're young, or even more so when you're born. So, we know that, for example, not work I've done personally, but, as you come through the birth canal, that's your first exposure to microbes, and that's your first on the way out. And then you're exposed to the outside world. And so then from there, and we know that if you're born via vaginally or cesarean section, there's a very big difference in what your microbial community structure looks like. I think in terms of saying that there's a relationship then with that and then disease, I don't, again, not work I'm doing personally, I don't think we're necessarily there yet, because a lot of these, there are two things that you really need to do in order to determine that. One, you have to track these individuals basically for a very long time, right? You have to see if at some point in their life they develop such disease. And obviously that's extremely difficult to do. So again, hard data to get, but I think people are trying to do this because you're exactly right. It's people believe that that particularly when we're talking about issues with inflammation in the immune system are really important. I mean, when we look at since the 1950s, we've seen this this steady increase in diseases that are of chronic origin, diseases that have an inflammatory component. So now we're treating a lot more chronic diseases than we are those one pathogen diseases that were, being treated 50, 60 years ago.
I got you. So what do you, I don't know, how would you figure out, for instance, the functionality of the different microbes in, a creature's microbiome? Like, how would you tease that out?
Yeah, so two ways. So we have multiple different types of sequencing that we can do. So one of the types of sequencing that we can do is 16S sequencing. So this is a ribosomal RNA gene. And what that basically means is that it's highly conserved amongst bacteria. This is not going to tell us anything about function, but this is the way kind of microbiome started. It's still used. It's still important, but certainly not as powerful as shotgun sequencing or metagenomic sequencing. With 16S sequencing, basically it will tell us who's there. The two issues. One issue is that it doesn't tell us what they're doing. Number one. Number two, it in terms of resolution, it only gets us down to genus level if we're lucky. The thing about bacteria is that species matter, and then even strain matters. And so, using metagenomic sequencing will do two things, particularly if the sequencing is deep enough. So this is shotgun sequencing. This type of sequencing will basically give you that resolution down to species or strain level in some instances, but it also tells you about microbial function. And then the other way is that we can use metabolomics, which is basically telling you what metabolites are being produced by the microbiome.
Yeah, have any microbes been profiled to the max? What is it? I don't know. E. coli. What does it eat? What's its genes? What does it produce in terms of metabolites? It's on and on and on. Like, I would think that would be very important because you could find who's there in gory detail, but then, okay, how do you know what they're doing?
Yeah, I mean, absolutely. Yeah, for sure. That's certainly been done with a fair number of bacteria. I think the thing is that if you think back, when you're talking about traditional microbiology, so culture-based techniques, a lot of these bacteria that have been worked with and characterized so extensively, like E. coli, for example, they grow aerobically. And so you have to figure out what type of nutrients they need and all these things to be able to go and study them in great detail. But it's a lot easier than a lot of the microbes that are in our guts, because those microbes are anaerobic in nature, and you need very, very special equipment in order to study those bugs. But you make a very good point, and I think that's the direction that the microbiome research is heading. At least I certainly hope so. Our lab is in that direction. Is that we have to characterize these bugs. So yes, we can. In order to do that, to fully characterize them, we have to culture them. So we can look at them taxonomically. We can look at what their function is, but to really truly understand. Let's say like we're screening for probiotic targets or whatever, we really have to culture these bugs. And the other thing about that is that databases. So databases are a huge issue. So if I go and today in a year from now and I blast something against the database, so I take a DNA sequence and I'm trying to figure out what that organism is, many times it'll come back with, you have, this percentage of unknown. And maybe if you redid that a year from now, because new things have been added to the database, you would be able to determine what actual species, let's say, that is. But in order to do that, that bacterium would have had to been cultured, its DNA would have had to been sequenced fully, and then, so that it could be compared to all the other species to make sure that it is a, novel organism.
But then what if the, I don't know, bacteria A and bacteria B act in one way separately when cultured, but when they're together, one feeds the other, which feeds another.
Oh man, that's a very, very, very great question and a very good point. I mean, that's obviously a limitation of in vitro work for sure. But you make a very good point. That's another thing that's complex about studying the microbiome, because you're not talking about one particular thing. You're talking about something in the context of everything else. And so yeah, there are different types of co-culture systems. There's instances where people are working, one in, one out. So you're adding, various different species and trying to understand the interaction. But this is work just like people are still studying E. coli like they always have, because there's still so much to learn, and that's just one bacterium. So I feel like it will take a lot of time, but there'll have to be a lot of people working in that area to elucidate exactly what you're talking about.
Yeah, I mean, like you think about, the skin microbiome. The stuff that's there is in a constantly changing, very adversarial environment versus, let's say the microbiome around my heart or something, or, in my brain, inside the blood-brain barrier. Mm-hmm. So I wonder if you looked at the different species of microbes in those two arenas, I would think they would be radically different. But then now I wonder, what if I had the same microbe X in both areas, and it had the same microbe in both areas? It may act so differently based on its environment that, I don't know, it would have abilities that it wouldn't have if it's in one area versus another. You know what I mean? Like, again, if I had microbe X and it was on my skin, but it was in a very protected area of my body, it may exist in both areas, but it may have very different roles and function and gene expression, and I don't even know. It's crazy.
I mean, that's absolutely correct, and there's numerous examples of exactly what you're talking about. I mean, you take Staph Aureus, for example. I mean, if you took I mean, staph, is inherently on your skin. You get a cut, and it's in a different part, then it results in infection. Or there's a number of different species that, let's say, for example, a really good example actually is E. coli. That's probably a real landmark example or whatever, because E. coli is from the gut. We know it's from the colon, but it's the number one cause of urinary tract infections in women. So E. coli is not supposed to be in the urinary tract. It's absolutely fine as long as it's not obviously a strain that's pathogenic, but it's a very normal part of the flora. That's why it's so easy to culture. But we know that, and it's there's nothing wrong with it. It's absolutely fine. But once it is in the urinary tract, it causes infection. So that's an example of exactly what you're talking about.
I don't know how this is going to be figured out.
It's going to take a long time, and a lot of people. And the one thing that is beautiful about microbiome research is it's highly collaborative, and the reason why is because it has to be. I mean, you have microbiologists, you have physicians, you have veterinarians, you have microbiologists, you have modelers, you have chemists, there's so many, because there's so many things that are going on, and you really have to have this breadth of I guess expertise or interest or knowledge or whatever in order to really kind of solve these complex problems. So it becomes a collaborative science, which is one of the things I like about it.
But what does that tell you about the capabilities of even microbes, of bacteria, of viruses, of even their behavior is so diverse. They act differently, and they're altered by their environment, and they have different things that they prefer to eat and will eat, and different things that they express, and they interact differently with different neighbors. I mean, that behavior is so complex even at that level. It's amazing. I have a new respect for bacteria.
Oh, absolutely. Yeah. I mean, that's why there's people that there are people that try to basically model this using mathematical models and stuff to prediction algorithms to try to predict what would happen, when you have these three species together, or four or five, but each time even when you're in, obviously this is a model, but even when you're modeling this, like think about how many, like exactly what you just talked about, how many factors you have to try to account for. And these models themselves are going to take years and years and years to refine, because it's so complex, and every time you add a new thing to the system, it's a whole nother level of complexity and change and potential outcome.
So it's hard not to feel overwhelmed. What's going to be your focus? How do you think you're going to add your piece of the puzzle? What do you think is the most important work that you're going to be doing or are doing?
Yeah. I mean, we're kind of the lab has a few different directions. So one of the things that we're focused on is using non-human primates as a model for studying the effects of variations in diet, as well as other lifestyle factors on the microbiome and metabolic health. That's one thing we're working on. But one of the ways that we're doing that is using this marmoset model, because having a model that we can use, and everything that we're doing is non-invasive, and so because we're using fecal material, so it works out really well in that regard. But it's the same with mice. So having a model that you can use gives you a lot of ability to look at stuff, as opposed to, let's say, using wild animals. But at the same time, there's a lot of stuff that can be garnished from that. So another thing that we're working on, really passionate about, is the Primate Microbiome project, something that started in a while ago with my collaborators at University of Minnesota, Northwestern University, University of Colorado Boulder, University California San Diego, and then University of Illinois Urbana-Champaign. So this project is the main goal of this project is that we want to basically to develop a systematic map of what the microbes are basically across the primate taxonomy. So, and part of the reason for that is because we want to collect and sequence the gut microbiome communities from primates covering the entire tree of primate evolution. I mean, we have a couple different areas of impact: health, evolution, behavior, and conservation. one of the things to me that's really important, and particularly when I think about from a veterinary perspective, and when I think about captive animal health, and when I think about zoos, and when I think about extinction, is that two things. One, we think about extinction most of the time on a macro scale. And so, in something that we can see, but we forget that microbes are highly host specific. And so when we're losing, let's say, a species, or even let's say a species in a particular environment, let's say pristine forest versus, degraded forest. And we know from studies that a lot of times in those instances, those two, let's say, same species of primate or whatever species have different microbiomes. And so we know that we would assume that pristine environment, once that's gone, yes, we may, let's say we lose that species, that host or whatever, but we're also likely losing bacteria that were very specific also to that microenvironment. And so one of the things that we want to do with this project, and why we want to understand what the microbiome looks like across basically all these primates is for conservation purposes. So we want to be able to help these individuals, for example, if they find themselves in a tricky spot or in captivity. And we know, in accepting the fact that how important the microbiome is for the maintenance of health, we want to both characterize these bugs and then also starting to culture these bugs so that we will, be able to both understand them better, but then also, and so they're available to science, but also so that we can use them for health purposes. And so that's another big aim in the lab, and that's obviously a highly collaborative project. And then the last thing that we're working on, which we're really excited about, is we're trying to explore detailed causal mechanisms for microbiome modulated metabolic diseases. So as well as neurological behavioral diseases. So specifically stress. So we're very interested in the gut-brain axis, and we're trying to understand the link between stress and other neuropsychiatric diseases in the microbiome. And we're doing that using like next generation sequencing, anaerobic, aerobic culture, germ-free models, and as well as that marmoset model.
I was just thinking, it'd be interesting. You're looking at diet. Why don't you try to see if you can feed some of the marmosets you have in captivity a wild diet, where the only food is not just variations of, human touch stuff, but anything they'd eat in the wild, and see what happens to their microbiome then. Does it go back to resemble their wild environment, or does it form this third type,
great
microbiome? What happens?
That's a great point. That's a very, very great point. Yeah. I mean, in some, so I absolutely agree with you on that. And I liked what you said about, does it revert back to the original, or is it something completely new? Or that was really cool, because I agree with you. Like you have a couple of different scenarios there. And I think one of the things that you think about is that how does it, for example, one of the questions that I have is that how long does it take, and do we have extinction events in the gut? So in other words, you have a monkey in captivity. Yes, it may have a different microbiome, but do you have some of these species still residual or in very low numbers that, let's say with the right substrate, they could rebloom, or you could basically increase their numbers, or are they completely extinct? So you basically they have to be re-entered into the microbiome in order to be a player, if that makes sense. So yeah, I absolutely agree with you. And in some instances, let's say, for example, the red shank duke, that's what I focused on, or one of the species I focused on for my PhD. In that particular instance, this species eats primarily leaf material, and it eats such a complex diet. That's probably one of the reasons they haven't been able to keep it in captivity very well, because the diet's so complex. It's just almost impossible to replicate. I like to use the example of that, let's say, versus a panda bear, where we know exactly what they eat. And you might try to formulate a diet, but you would expect they're not going to do very well. And so what you grow their food, you have these farms to grow their food. But with some of these individuals that, are in the rainforest and eating, 100, 150 different types of, and many of which are not even known, because if the species hasn't been studied, then that makes it complex. But I absolutely agree with you. In the case of one of the things that's interesting about marmosets specifically is that they're called gumivores. I mean, they're technically omnivores in the sense that they eat a wide range of food just like us. But one of the things that's unique about them is that they actually eat gum. So they eat from trees. And so that's and that's likely something that probably dramatically affects their microbiome, and that's something that we've actually already begun to see. But yeah, 100% agree. Posing a lot of good ideas and questions. So this is very thought provoking. I'm almost thinking as I'm talking.
Oh, good.
Yeah.
Well, good. That's excellent. Yeah, I mean, I would think that, what if you assume that everything affects your microbiome, and then your approach is to find the things that affect it most strongly and make it like a Pareto, like an 80-20 analysis of what affects it most strongly, and then focus on the things that affect it most strongly to gain insight, or most weakly to gain insight. I don't know. What if you
go ahead,
pick a part of the creature you're looking at that has the simplest microbiome by its nature, maybe not the gut because it's so complicated, and maybe by looking at the simplest part of the creature, I don't know, it's eyelash or something that has very few species there, maybe then you could figure out what happens with the host microbe interaction. Look at it in a simple way.
I think, yeah, I mean, exactly. I think you're nailing it there. And even I think another thing that's important, even if we're looking at the gut, let's say, I think the way that and that's why, let's say even with humans, that's why it's difficult. Because if we think about, if you were to write down what you eat every day, it would be astonishing how many different things most likely that people eat. But not even that, when you look, particularly in westernized society, when you look at the components of what's in those foods, I mean, it's a laundry list of things, right? And so trying to identify what's causing what is difficult. That's one of I think the advantages of working in the lab is that we can, not everything, of course, but we can control many things. So if we can do every single thing the same with, let's say, with a group of primates or mice or whatever, then we can start to then try to define a difference that's meaningful because of the fact that everything else is kept static.
Yeah. Well, very good. There's 8 million more questions, but there's only so much I can do before our brains explode. So
right, absolutely.
So what's the best way for people to get in contact, with the lab or the institution, or to ask questions or see papers that you're putting out?
Yeah, absolutely. So it's Jonathan B. Clayton on Google Scholar. So there, another place is the University of Nebraska at Omaha in the Department of Biology. And then at the University of Nebraska-Lincoln, the Nebraska Food for Health Center is a place also where a lot of that information exists. Another place is the Primate Microbiome Project. We'd love for people to check it out. And anybody that's interested, there's actually a link on there for an email link. So anyone that's interested, we'd love to hear from you with questions, or if you're interested in collaboration, we would love to talk to you. So I'm always open for people that are interested in checking out the lab, that people want to work in the lab, that are interested. We encourage to contact me and see what we can do.
Well, that's great. Well, Jonathan, thanks so much for coming on the podcast. I appreciate it.
Yeah, thank you very much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.